my scientific poster, I did the Hill Reaction Lab, and my group used the collard greens. So, thylakoid membranes are affected by different treatments. Over on the left, you see my abstract section where I talk about how collard greens harvest energy from the sun to make energy for themselves using CO2 molecules that surround them. Thylakoid membranes are where photosystems are found, which absorb and transfer light energy, also known as transferring electrons that are needed for photosynthesis to occur. We isolated those membranes from a collard green leaf in order to collect data on the absorption of electrons in different conditions, which essentially is measuring photosynthesis. This lab utilizes photosynthesis, and we must ask ourselves, why is photosynthesis important? Well, plants are autotrophs, so they make their own food, and they use photosynthesis to transform water, sunlight, and carbon dioxide into oxygen and sugars that the plant will eventually use as fuel. A number of steps must be taken in order to isolate the thylakoid membranes, as we see in the top middle for my methods. First, we must make sure that we've removed the mid-vein from our leaf. Then, using a mortar and pestle, which you see to the left of the methods, you grind the leaf with 10 milliliters of grinding buffer. Once a paste is formed, you add more buffer, then squeeze that paste through a cheesecloth. From there, using the liquid obtained, you use a centrifuge twice in order to obtain a pellet which would contain our thylakoid membrane. Then, taking that pellet, you resuspend it in more buffer. At this point, we have isolated our thylakoid membranes from our collard green leaf. As seen in Table 1, we must divide the membranes into different tubes for each treatment. Table 1 shows the reaction tube's ingredients. We added water, 130 microliters of DCPIP, reaction buffer, herbicide to some, and of course our chloroplasts. Once we have obtained each separate tube, total of 7, in 10-minute intervals after sitting in the sun, we measured absorbance using a spectrophotometer. Now over on the very right, you can see our results. The first graph shows the absorbance at 600 nanometers versus time in minutes of the first 4 treatment tubes that are listed in Table 2. For the first graph, the yellow line represents chloroplast plus herbicide treatment. The blue line represents no chloroplasts, red line represents chloroplast plus dark, and the gray line represents chloroplast plus light. Second graph shows the absorbance at 600 nanometers versus time in minutes of the yellow and red covered tubes. For the second graph, the blue line represents the yellow film covering, and the orange line represents the red film covering. For both graphs, we can see that over the 30-minute interval that we measured absorbances, the absorbance decreased as time went on. The results show that each different treatment had an effect on the absorbance of electrons done by the photosystems. The overall mean absorbency, which is shown in Table 2, for the chloroplast plus light tube was the lowest among all the others, meaning it accepted the most electrons. This makes sense because in order for photosynthesis to occur, light energy is needed. Since our chloroplast plus light tube was the only tube that is essentially how normal photosynthesis occurs, meaning that there was no other factors like herbicide or a yellow film or dark added to that tube, it would showcase photosynthesis. The tube containing herbicide had the highest absorbance reading, showing that herbicide had an effect on how many electrons could be accepted. Making sure each tube had access to the same amount of sunlight and had a consistent way to cover the dark and colored treatment tubes would help our results be more accurate in the future. The act of taking off the tinfoil and the films in order to measure our tubes every 10 minutes could have skewed our results because of the potential human error in the inconsistency of placing the foil and film back over the tube. Overall, I think this was a very successful lab, and it was definitely one of my favorites that we did this semester. It really helped tie in everything that we've been learning in our regular bio class, and seeing it happen in a lab is definitely much more interesting than just learning it in class. Seeing how we can actually take a leaf and isolate its thylakoid membranes to see how photosynthesis really works is really helpful in the understanding of photosynthesis.